Hello, my name is Merrick from the University of Adelaide, and this project looks at characterizing the influence of cerebellum on the neuroplastic modulation of intracortical motor circuits. During motor learning, we recruit many different regions in the brain and produce changes in the strength of synaptic communication between neurons, which is a process referred to as neuroplasticity. When we consider neuroplastic events in the context of motor learning, the communication between the cerebellum and the motor cortex is crucial. Specifically, the cerebellum seems to process sensory feedback during motor performance in order to update commands in the motor cortex. However, the neuronal elements within motor cortex which are targeted by the cerebellum is currently unclear. One of the many useful ways of assessing these connections is to use transcranial magnetic stimulation, or TMS. When applied over the scalp above the motor cortex, TMS emits an electromagnetic pulse which activates the motor neurons below it, activating the cortical spinal tract, thus producing a motor evoked potential, or MEP, in target muscles. It is thought that a change in cortical spinal excitability, or a change in the MEP, reflect neuroplastic events. So naturally, TMS has also been used to assess neuroplastic events. And studies have identified a particular set of intracortical networks within motor cortex called I-wave networks, which seem to be important for neuroplasticity. When a single pulse TMS is applied over the motor cortex, it actually recruits I waves, and there are different types. It recruits early I waves, as shown here in green, followed by late I waves, shown in red. And the activity of these I waves then summate at the spinal cord, producing the MEP in the target muscle. Interestingly, it is also possible, or studies have also shown that it is possible to selectively increase the excitability of early or late I waves, which would subsequently increase the cortical spinal excitability. One of the techniques used for this is a repetitive pair pulse TMS technique called I-wave periodicity TMS, or ITMS. And this protocol pairs two TMS pulses together at an interstimulus interval in ISI, corresponding to I-wave periodicity of approximately 1.5 milliseconds. Subsequently, an ISI of 1.5 milliseconds targets early I waves, while an ISI of 4.5 milliseconds target late I waves. The key point being that it is possible to modulate or to modify these TMS parameters in order to selectively increase the excitability of early or late I waves, and therefore increase cortical spinal excitability. However, it is unclear if the cerebellum influences I-wave plasticity. What we do know are that late I-waves in particular are involved in error-based learning, which seem to heavily engage the cerebellum. Furthermore, modifying cerebellar excitability with direct current stimulation appears to specifically alter late I-waves. Taken together, this suggests that cerebellum likely modifies late I-wave plasticity. And hence the aim of the project was to assess how downregulating cerebellar excitability influences the neuroplastic response of different I-wave networks within motor cortex. And we hypothesized that modulation of the cerebellum would specifically influence late I-waves. For our methods, we assessed the effects of downregulating cerebellar excitability on early and late I-wave plasticity in 22 healthy young adults. Each subject participated in 3 experimental sessions on separate days in which the excitability of the cerebellum was downregulated using cathodal transcranial direct current stimulation, or cathodal TDCS. In addition, we applied concurrent ITMS to target the plasticity of early I waves using 1.5 milliseconds ISI, late I waves using 4.5 milliseconds ISI, or a control session in which we use varying ISIs that produce no change. The key point is that we used an intervention that allows us to see how modulating the cerebellum influenced the plasticity induction of different I waves. For baseline and post-intervention, we assess cortical spinal excitability using single pulse TMS, as well as cerebellar excitability using cerebellar brain inhibition, what's called CBI. CBI involves a conditioning pulse to the cerebellum followed by a test pulse to M1, which produces an inhibited response, thus allowing a measure of cerebellar inhibition. All recordings were made from the first dorsointerosseous muscle, the FDI, and post-intervention measures assessed at 5 and 30 minutes were normalized to baseline values for analysis using repeated measures of linear mixed models. And using this experimental protocol, we found that there was a significant main effect in session for cortical spinal excitability. Looking at figure 5 here, which illustrates post-intervention responses of single pulse MEP following ITMS 1.5, 4.5 in sham, you can see that responses following ITMS 4.5 and ITMS sham were reduced compared to ITMS 1.5. More interestingly, the facilitatory response, which is typically expected following ITMS, is absent following ITMS 1.5 and 4.5, suggesting that this facilitatory response is removed when coupled with a cathodal cerebellar TDCS, suggesting a generalized reduction in I-wave excitability with cerebellar modulation. More importantly, the extent of this response is greater following ITMS 4.5, suggesting perhaps a stronger influence of cerebellum on late I waves. We also found a significant interaction between session and time for cerebellar excitability. Figure 6 depicts the CBI responses following the 3 ITMS conditions at 5 and 30 minutes. You can see that following ITMS sham, there was an immediate reduction of CBI at 5 minutes. Also, this was the case for ITMS 1.5 and 4.5 at 30 minutes. But the point here is that there was disinhibition across all conditions, indicating that cerebellar TDCS reduced cerebellar excitability. So from this study, we can conclude, well, it suggests that concurrent cerebellar TDCS and ITMS result in motor cortical dysfacilitation, with a stronger effect shown in late I waves. However, our findings of reduced cortical spinal excitability suggest an overt cerebellar modulation, whereas our findings for CBI suggest a reduced cerebellar inhibition, and therefore further examination that more precisely disentangle the effects of cerebellar TDCS and ITMS are required.